For over a century, the story was settled. Neanderthals were a pure European lineage, forged in Ice Age isolation. Then scientists extracted DNA from bones that were 430,000 years old, found inside a Spanish cave, and they discovered something impossible. Fossils that looked unmistakably Neanderthal carried Denisovan maternal DNA and a Y chromosome closer to modern humans. Both lineages were overwritten. To understand how ancient DNA shattered everything, you need to start with the story everyone believed. In August 1856, laborers digging for limestone in Germany's Neander Valley uncovered bones that no one could explain. Schoolteacher Johann Karl Fuhlrott recognized they were ancient, and anatomist Hermann Schaffhausen confirmed their human origins. The skull's heavy brow ridges and thick vault set it apart from anything in Europe's record. Within a decade, the bones had a name, Homo neanderthalensis, and a story that would shape generations of textbooks. The classic narrative took shape as more fossils surfaced across Europe. Neanderthals, the story went, were the direct descendants of Homo heidelbergensis, an early human ancestor that spread from Africa into Europe roughly 600,000 years ago. Around 400,000 years ago, this lineage supposedly split off and settled across Ice Age Europe, cut off from the outside world by glaciers and distance. Over the next 350,000 years, Neanderthals were thought to adapt in isolation, forging a body built for cold, broad noses, stocky frames, and powerful limbs. Their Mysterian stone tools, unique to Europe, reinforced the idea of a people apart. For most of the 20th century, this vision held steady. The Neanderthals were cast as a static, pure European branch, an evolutionary dead end. Only around 45,000 years ago, according to the textbooks, did their world change. Anatomically modern humans, Homo sapiens, arrived from Africa, bringing new tools and art. The two groups overlapped briefly, perhaps exchanged a few genes, and then the Neanderthals vanished. Their story, it seemed, was a closed chapter, a long, cold isolation, a fleeting encounter, an extinction. Yet, as more fossils turned up and radiometric dating grew sharper, cracks began to show. Some sites hinted at earlier modern human presence. Debates flared over the true identity of their ancestors and the length of their isolation. But the old model endured, printed in museum displays and school books, a neat, linear tale of a people apart. The question lingered, was this story ever complete? Or was something fundamental missing, waiting to be revealed by evidence still locked in ancient bone? Deep beneath the limestone hills of northern Spain, a narrow shaft plunges 13 meters into darkness. This is Cima de los Huesos, the pit of bones, where more than 24 ancient humans met their end about 430,000 years ago. For decades, the site was a puzzle. So many individuals, their bones scattered and mingled, preserved by layers of calcite and time. Yet, their skulls and jaws showed features that seemed to bridge the gap between earlier ancestors and the classic Neanderthals found across Ice Age Europe. In 2016, a new line of evidence arrived in the form of ancient DNA. Matthias Meyer and his team at the Max Planck Institute set out to recover genetic material from these fossils, pushing the limits of what was thought possible. The process demanded extraordinary care. Each bone was handled in a clean room, surfaces decontaminated, and even the air filtered to prevent modern DNA from clouding the results. From the dense core of a femur, they coaxed out fragments of nuclear DNA, genetic instructions inherited from both parents, locked away since the Middle Pleistocene. Sequencing this nuclear genome, the team found a clear pattern. The Cima de los Huesos individuals clustered tightly with later Neanderthals, like those from Vindija and the Altai. The genetic signature was unmistakable. These were early Neanderthals, already on the branch that would define the species for hundreds of thousands of years. The results seemed to validate the textbook model, Neanderthals evolving in Europe, their story stretching back to these very bones. Confidence soared. But another test, probing a different genetic compartment, was about to tell a stranger story. From the start, the Cima de los Huesos DNA project was a gamble against time and decay. Ancient DNA fragments this old, 430,000 years, are usually shattered beyond use. Yet, following every protocol, The Max Planck team coaxed a nearly complete mitochondrial genome from a single tooth. Mitochondrial DNA is inherited only from mothers, passed down the maternal line like a family heirloom. It acts as a genetic time capsule, tracing ancestry through the female side. The result, when it arrived, was a jolt. Instead of matching later Neanderthals, the SEMA mitochondrial DNA clustered tightly with Denisovans, a mysterious branch of ancient humans known only from Siberia. This made no sense. The bones looked Neanderthal, the nuclear DNA said Neanderthal, but the maternal signature pointed east to Denisovans. The team scoured their methods for errors. Every extraction was run alongside blank controls. Patterns of DNA damage matched ancient origin, not modern contamination. Less than 1% of the sequence could be traced to modern humans, far too little to explain the result. The evidence held. Somehow, a population in Ice Age Spain carried a Denisovan-like maternal legacy. Could Neanderthals have absorbed Denisovan mothers, or was this a ghost signal from a deeper, tangled past? The paradox forced a new question. Just how permeable were the boundaries between ancient human groups? In 2018, a single fragment of bone from Denisova Cave in Siberia upended the last defenses of the old story. Labeled Denisova 11, the specimen looked unremarkable, a sliver of long bone, thousands of years buried in the cave's cold earth. But when Viviane Sloan and her team at the Max Planck Institute sequenced its DNA, the results landed like a thunderclap. This was no ordinary fossil. The genome split cleanly down the middle, half Neanderthal and half Denisovan. Denisova 11 was the direct daughter of parents from two different ancient human groups. Her mother's DNA carried the telltale markers of Western Eurasian Neanderthals, while her father's genome was Denisovan, with traces of earlier Neanderthal ancestry woven in from even older encounters. The implications were immediate and profound. Here was living proof that Neanderthals and Denisovans not only overlapped in time and space, but mated and produced fertile offspring. The walls between lineages, once thought to be absolute, now looked more like porous borders. The 50-50 split in Denisova 11's genome made the idea of maternal or paternal replacement in Neanderthals not just possible, but likely, even routine. For researchers, the hybrid's DNA was a smoking gun, direct evidence that boundaries between ancient human groups were crossed again and again, leaving genetic traces that still echo in our genomes today. The next blow to the textbook story arrived in 2020, this time from the paternal side. The Y chromosome, passed only from father to son, acts as a genetic record of male ancestry. For years, researchers assumed Neanderthals carried their own distinct Y line, isolated like the rest of their genome. But when Kate Proofer and colleagues sequenced Y chromosomes from Neanderthal fossils dated between 150,000 and 100,000 years ago, the results scrambled expectations. Instead of clustering with earlier Neanderthal lines, these Y chromosomes, grouped with a branch found in modern humans, haplogroup CT predating the split of most living Y lineages. The turnover was abrupt and widespread, appearing in Neanderthals from both Europe and Siberia, and it happened tens of thousands of years before the main wave of Homo sapiens moved out of Africa. The timing forced a reckoning. The replacement could not be explained by the late, fleeting encounters described in old models. It pointed to an earlier episode of gene flow, when small groups of early modern humans, or perhaps their close relatives, entered Neanderthal territories and left a lasting paternal mark. Some geneticists argue the new Y chromosome swept through Neanderthal populations because it offered a survival advantage, perhaps tied to immunity or fertility. Others suggest chance played a role. Either way, the evidence was unmistakable. Neanderthal paternal ancestry had been overwritten by an outsider lineage long before their final encounter with Homo sapiens. The idea of an untouched, isolated Neanderthal genome was unraveling, replaced by a story of repeated contact and systemic rewriting at the very core of inheritance. Neanderthal bones hold a paradox. Their bodies, shaped by thousands of years in Ice Age Europe, kept the same broad noses and powerful limbs even as their genetic heritage shifted beneath the surface. Ancient DNA reveals a story written and rewritten. Denisovan mothers left their mark through the mitochondrial genome, while early modern human fathers replaced the original Neanderthal Y chromosome. Yet, the outward Neanderthal form endured, preserved by natural selection, favoring traits best suited for survival in harsh climates. This is a genetic palimpsest, layers of ancestry overwritten, but the physical script of Neanderthals remains legible, blurring the line between identity and inheritance. Denisovan fossils remain almost invisible, just a handful of teeth and bone fragments from a cave in Siberia, plus a jaw from the Tibetan Plateau. Yet their genetic fingerprint stretches across continents. Today, people in Papua New Guinea and Australia carry up to 5% Denisovan DNA, while traces appear as far as East Asia and parts of Oceania. This reach is astonishing given the scarcity of physical remains. The Altai research team, working with so little material, uncovered a genetic legacy that outpaces the bones themselves. Denisovan genes flowed far beyond their known fossil range, showing that ancient populations were anything but isolated. Even with only fragments to study, the evidence points to a prehistoric world shaped by connection, not by walls. The old story of a pure, isolated Neanderthal lineage no longer stands. Ancient DNA has exposed a legacy written and rewritten by outside influences, Denisovan mothers, early modern human fathers, and gene flow that crossed continents and epochs. Yet, crucial questions remain. Most of the ancient genomes come from Western Europe and Siberia, leaving the Levant and Central Asia as blank spots on the map. High coverage Y chromosome and mitochondrial DNA from these regions could reveal missing chapters in the Neanderthal story. As new technologies push the limits of what ancient bone can tell us, the debate over where one species ends and another begins grows sharper. The search for Neanderthal origins is far from over. Each discovery only deepens the mystery of what it means to be human. Today, ancient DNA exposes Neanderthals as a genetic palimpsest, written, erased, and overwritten by migrations we are still decoding. Their story dissolves the myth of purity in our own origins. As science redraws the boundaries of species, it leaves us with a deeper question. Where does one lineage truly end and another begin?